First, thank you, Dr. Ramaswamy, for allowing us to have this interview with you. It's very nice to meet you. Can you please tell us about your journey from college to your PhD and all the work that you're doing right now?
So my undergrad was in bioengineering, which is the same as biomedical engineering pretty much. After that, so once I graduated with a bachelor's degree, I initially was very keen on going to industry and working. So I did a few I guess odd jobs here and there, and nothing too permanent for a while. And then finally I ended up getting a position in the university as a research engineer type position. So I was working full time and said, okay, while I'm still here, maybe I'll improve my resume and do a master's degree part time. My master's was actually in material science. It was with biomedical application. So it's kind of like stepping out of biomedical and doing more materials engineering, but the application was biomaterials. So, but having done that, I realized that the BME field was very research driven, and now don't forget you're talking about this was like the late 90s, where a lot of industry BME was still developing. It wasn't really there yet. I mean, there were companies hiring BMEs, but it was still a time when a lot of people didn't even know what BME was. So just as a matter of sheer coincidence and good fortune, there was a group from France who were very interested in the work that I did in for my master's, and they asked me if I would be interested in spending a year doing an internship for one of the national labs in France, and they would pay for the whole thing. So I was like, sure. I mean, that's great. So fast forward, I spent 5 years at Iowa. Completed my PhD. PhD was in cardiovascular mechanics primarily, but I was always interested in the stuff that I did during my master's, which was partly tissue engineering, partly material science, partly musculoskeletal applications. But it was really the cell and tissue engineering component that I felt like at some point I need to revisit that. So after I was done with NIH, I looked at my skills with computational modeling, with MRI, with material science, and I figured that I wanted to really apply all of this towards the cardiovascular area, because I saw that there was opportunity there. There really wasn't as much being done in the cardiovascular area. The only reason really I decided to pick an academic job, and this is just the honest answer, is academia valued me more than industry. So I was getting better offers. I seemed to be a better fit, and never really thought about academia, but I guess life has a funny way of giving you something that you don't think you're really I never really thought that I would ever be qualified enough for becoming an academic, because you always have this stereotype that as a professor you need to know a lot. And so I was like, okay, my research here primarily focuses on cardiovascular regenerative therapies, and a large part of that for the last 8 years, like I said, has really focused on critical congenital heart valve disease. We approach that problem with several engineering tools: modeling, tissue engineering, model systems, biomaterials. So all the stuff that I actually learned, as I mentioned to you, all the stuff that I actually picked up, we're using those skills in the lab. And I think that's what it really is about. It's about awareness of bioengineering tools, that you have a good handle on how to use these tools, and then taking a really multidisciplinary approach to trying to solve a problem. So one of the exciting new projects that I've really been moving on right now is in the area of stem cell therapy for cardiac repair. So not just looking at heart valves, but actually the whole heart, and specifically looking at how cell therapies can improve cardiac function. And so the main focus there is to really take some bioengineering approaches to improving and optimizing the therapy of stem cells, because right now for a large part stem cell therapies are still very much a aim, shoot and see approach, and there's value in that, but after several animal studies and human trials, there's evidence that there's significant improvement with the use of certain types of cell therapies. But I think with bioengineering approaches, we can make those therapies that much more repeatable and make those outcomes more effective and actually overall improve the therapy itself. And so there's a number of ways that that can be done. And again, we focus on modeling, we focus on mechanics, we focus on cell and tissue culture, and these type of experiments will help to objectively improve cell therapy. So that's kind of like the new kid on the block, the new area that's in the lab. And so we're hoping that that would be one of the main research themes in the lab. And of course, our bread and butter is still heart valves. And we have two main projects within the heart valve area. One is using stem cells to grow repair tissue for valves. And the other one is a pretty exciting project looking at porcine small intestinal submucosa, which is basically fancy words to say tissue from the small intestine of pigs, which is cleaned up and then subsequently used as a scaffold for repair and replacement purposes. And we currently have an AHA-funded project looking at how these porcine tissue scaffolds can be used in mitral heart valve replacement. And we've actually gone ahead and implanted some of these valves in baboons. And so one of the exciting things is we're beginning to see that there's some evidence of growth, that these are young baboons, and as they grow, we are noticing that the valve is also growing with them. That really can revolutionize treatment of valve disease in children, because for children there isn't a appropriate valve that they can adopt because of sizing limitations. Kids are very small, and of course as kids grow, the valve doesn't grow with them, because right now there's really no good options for kids with serious valve diseases. Of course, as bioengineers, our job is to see how we can make this treatment better. I mean, I think it's definitely a very good time to be in science. I think a very good, exciting time with all the technology breakthroughs that are happening right now with gene editing, and it's all over the news. It's a very exciting time to be in science, but at the same time, science alone and thinking of science alone is not sufficient to be successful in a research career, because there are so many other factors: legislative, financial, regulatory, and so you want to be sure that if you are interested in research, which is I think a very rewarding career, but be prepared to wear several hats. So I think the advice I would give to people is try to stay focused, but also talk to people who are not in your field. Recognize all the issues, not just the scientific one. And of course, part of the role is, as you transition out of a bachelor's degree and move forward, if you're planning to take, go to graduate school or med school, then you have to also keep in mind that outreach and giving back is also very important. So you want to make sure that you are also, even at the undergraduate level, are in a position to serve as a mentor and have mentees, even to junior high or high school students, because it's very important to continue to inspire people. I think the inspiration that you get is critical to move somebody through this process. Maybe it's more important than the process itself.